OsteoBites is a weekly osteosarcoma webinar and podcast presented by MIB Agents. Today we're speaking with Andy Woods, research scientist, and Dr. Charles Keller, scientific director of Children's Cancer Therapy Development Institute. Our panelists are Shannon McCormick, OsteoWarrior Sibling and ambassador agent, and Amanda Levine, OsteoWarrior and patient advocate. And I'm your host, Anne Graham. Welcome to OsteoBites, everybody. Today, my bite of the day is kind chocolate, which is super appropriate because we are talking today about one of the greatest kindnesses possible. I hope you have your snack ready too for this OsteoBites. MIB Agents makes it better for kids with osteosarcoma. We help kids and families facing this aggressive cancer by providing direct patient and family support, educational programs for physicians and families by funding meaningful osteosarcoma-specific research. It is Childhood Cancer Awareness Month. We have for 7 more days, and we are $3,500 shy of our $40,000 goal to support our programs for kids and families in treatment for osteosarcoma. We have had incredibly high demand due to the global pandemic, which has suspended music, pet, and art therapy programs in hospitals, as well as limiting visitors for patients and patient families to one parent. Loneliness and isolation are very much a struggle for kids in treatment anyway. So we have been able to provide this meaningful and ongoing support and resources to the families because of you. If you're able to continue to help or start to help make it better, please go to mibagents.org/September. Thank you, thank you, thank you. Andy, would you get us started by introducing yourself, please?
Sure. My name is Andy Woods, and I'm a senior research associate at the Children's Cancer Therapy Development Institute just outside of Portland, Oregon. And at the Institute, I study Wilms tumor, several sarcomas, and then I also run our Cure Fast tumor banking program. I come to be interested in science due to my family's personal experience with childhood cancer. When my oldest daughter was 4, she was diagnosed with Wilms tumor, which is the most common kidney cancer discovered in children. And I'm happy to say that she's 7 years cancer-free now. But the experience was life-changing for all of us. And I sort of became very passionate about children with cancer, and particularly those that have rare cancers with not very good treatment options. And so I also was very interested in science and made a midlife career transition from building homes and setting tile to studying cancer. And so I've been at cc-TDI now for a little over 3 years.
So I'm Charles Keller, the scientific director of cc-TDI, and I could tell you a few things about cc-TDI. How do we try to bring scientific, basic science and translational research in partnership with the community to clinical trials for kids, adolescents, and young adults with cancer. But I think one of the most fun parts about today's talk is first Andy's story, where he came to one of our 1-week boot camps for childhood cancer, our NANA course, and asked, well, how can I stay? And the answer was, well, he got himself an internship, followed by enough preliminary data to get $280,000 to study his own form of cancer, but his daughter's own actual form of cancer. But more importantly, the project that he's going to talk about, a relatively sensitive topic about creating a small silver lining to the cancers that are hard to treat, is that this work is supported by some fabulous community foundations. And so I just wanted to do a shout out to the Sam Day Foundation that broadly funds the project, Maddie's Promise Foundation, which enables us to do additional patients with rhabdomyosarcoma, another type of musculoskeletal sarcoma, but of course the Childhood Cancer Project that is our main funder for osteosarcoma legacy gifts and research, so that the world will have the resources to understand the hardest to treat osteosarcomas. So with that, thank you very much, Anne, for this opportunity, and thank you, Shannon and Amanda, for being panelists at this presentation.
Hi, my name is Shannon, and like Anne mentioned before, I'm a sibling of an OsteoWarrior. My older brother Dylan is now 20, and he is 6 years cancer-free. And throughout his journey, I had become an ambassador for MIB myself and gotten involved in any way I can. And I just wanted to say thank you for having me today.
As always, I thank MIB for having me. My name is Amanda, and I've been involved with the OS Facebook groups for about 10 years. I had OS 33 years ago, breast cancer 10 years ago, and ALS now for the past 2 years. And today's a very important conversation we'll be having, and I'm honored to be here.
Okay, so my name is Andy Woods, and I'm going to give a talk today called Legacy Gifts, a research autopsy opportunity that gives back to families. So just a little background about cc-TDI, which stands for the Children's Cancer Therapy Development Institute. We're located just outside of Portland, Oregon, and we're a nonprofit biotech whose mission is to make all childhood cancers universally survivable. Our motivation and inspiration are children like Alexa, pictured here in the center. Alexa was an amazing person, incredible student, great friend, and an amazing daughter. And unfortunately, she had metastatic osteosarcoma. And we lost Alexa a couple of years ago now. But her and children like her are very motivational and inspirational to us. As many people on the call probably know, osteosarcoma is a cancer of the bone. It affects about 900 people a year in North America, and it has about an 80% cure rate. But unfortunately, when it's metastatic, meaning that the tumor has moved from the bone to other areas of the body, the survival drops down quite a bit, with perhaps less than 30% of those patients surviving. And so we at cc-TDI really want to improve those statistics and outcomes. One of the ways we do that is through our CureFast tumor banking program. And so this is a program set up and designed to collect tumor tissue for research purposes. And not only do we do research on the material that we receive, but we also develop study models. And when I say study models, I mean cell lines and mouse models, which are very useful for the preclinical research communities. And so the tissue that we receive comes in the form of surgical leftovers. And then also there'll be autopsies when a patient passes away from his or her cancer. And we can collect the tissue from those autopsies. And we call those donations legacy gifts. So these are painless gifts of tissue made after life that oftentimes fulfill the patient's last wishes. And of course, they're a valuable contribution to research. So why do we need this tissue and research and study models? There's a statistic out there that's been around for a while. 1 in 5 children with cancer will not survive. And so we would very much like to improve that statistic and make it so no children diagnosed with cancer pass from their disease. So in order to get to that point, more research is needed. And oftentimes with these very rare cancers in particular, there's just no study models. There's no cell lines, no mouse models. That leads to this problem where because there's nothing to study, researchers who are interested in those diseases don't have much to work with. And so the CureFast program is set up to design models to provide both to our own research at cc-TDI and to other research groups who are interested in studying rare cancers. One point to make is that even the cancers with existing study models, oftentimes those study models are old, and there's only a few of them. And in order to reach statistical significance, you need many models. And when I say old, some of these cell lines have been around for decades, and they've been passaged and worked, and may not even represent the original diseases that they were intended to. So there's a great need for more study models and newer study models. I just put this quote up there as I was preparing for this talk, and I was reading an article on osteosarcoma where they said that diversity of available cell lines is limited, and generally cells have been cultured extensively, not defined genetically or poorly genetically characterized. And the CureFast program is set up to address that problem. So just as an overview, the way that sort of the research pipeline works is it moves from basic science and target identification to proof of concept preclinical studies. So this is where we use those study models, and we basically ask ourselves a question, you know, if we find a target and then we can use drugs against that target, does that result in tumor regression or cures? And so once we've sort of addressed that at the preclinical stage, then we move into the clinical stage, where we partner with pharmaceutical companies and cooperative groups such as the Children's Oncology Group to run clinical trials in children, and hope that the preclinical work will lead to better treatment options and cures for those kids. So at cc-TDI, we have a special focus or emphasis on the preclinical side of things. So basic science, target discovery, and proof of concept. Then we partner to run clinical trials. And these clinical trials are great because a lot of times children who have gone through frontline treatment, then relapse, are not left with any further treatment options. And so clinical trials represent an option to try. And of course, in the best case scenarios, they work, and that leads to cures for children. The study models created in the CureFast program through surgical leftovers and legacy gifts inform the preclinical work. What is the process of making a legacy gift donation? It almost always starts with an introduction, and this is done by either doctors reaching out to us and mentioning that they have patients interested in donating tissue, or it's done the patients directly reaching out to us, or a lot of times it's done through other members of the community, the childhood cancer community, who know each other and know of situations and know of families that are interested in contributing to research. And so through that variety of ways, those people are introduced either to my scientific director, Dr. Charles Keller, or to myself directly. Once we have the introduction, I meet with the family and we talk about the CureFast tumor banking program and what we do and how sort of a donation would work. And if they're interested and want to pursue a donation, then we enroll them onto our CureFast study. And that's done with a simple consent form. Then we arrange for the autopsy in the case of legacy gifts. We'll talk more about that later on in the talk. After the tumors have been collected, then there's basically an overnight delivery in a FedEx box most often, and it comes to our lab. Once it comes to our lab, we go through multiple steps of processing that tissue. The lab is always bustling on the days that CureFast donations are coming in. There's a lot of activity. We want to be as prepared and ready as possible to start right away as soon as we receive the tissue. So we track it as the package is coming, and then the moment it comes through the door, everything's ready and we start right away. This is the workflow of what we oftentimes do with tumor tissue. And so it's divided up. We store some of it for future use. We store it in multiple different formats and ways. We develop cell cultures with the tumor tissue. And then we can take those cell cultures, validate them to make sure that they're tumor and not a normal sample. And then we can do a variety of tests with those cell cultures. One of which is pretty standard in our pipeline, is to do a 60 compound drug screen. And so we're already collecting data on this tissue, which might be helpful for living patients. The other side is that we implant some of the tissue into mice through our collaboration with Jackson Labs, and attempt to develop mouse models, which are available publicly through the JACS catalog. So any researcher that's interested in rare childhood cancers, oftentimes they'll go to JACS to look for mouse models. And we've built a lot of those models from the CureFast program. We also do a deep dive genetic sequencing analysis on the tissue, which is very useful and just gets more knowledge about those samples. And that, of course, is very useful for our own research, but it's also nice when we share the samples with other groups, which we proactively do. And not only can we provide, a cell culture or a tissue sample, we can also provide the genetic sequencing information, results of any assays that we've already done on the tissue. So it's really an informed way of sharing. Another way of sharing is just to publish information, and that just gets information out to the field. And so here is sort of a smattering of papers that we've published, some about autopsy itself and the process of legacy gifts, some which characterize, a PDX mouse model that we made. Oftentimes these mouse models are world's first for the disease. And of course we publish case studies as well. So if we have an interesting case, it's maybe a little bit unique, we'll write up the case, we'll do a deep dive into the genetics, and then we'll publish that and present it to the field. Another way to share is to actually share the physical samples. And so this was a picture taken just a couple of days ago, where we collected, I believe it was 79 tissue samples from 40 patients. And we sent them to the Children's Oncology Group Biopathology Center, which is the COG's tumor bank. It's a great way to share tissue because, we're a small lab in Portland, and we know a lot of people in the community, but sometimes people don't know us. And if they're looking for tissue, the first thing they'll do is go to the COG directly and ask. And so being able to share our samples with them is a great way of sharing to the worldwide community. This is just a list of some of the PDX models that we've made in the past with our collaboration at JACS. And like I said before, many of these are world's first. And so a good example of that is the epithelioid sarcoma, which until we had sent tissue to do the mouse model, did not have a mouse model that represented the disease. So once that was created, it was huge to move forward research for epithelioid sarcoma. As I mentioned, another thing we do with the CureFast tissue collected through legacy gifts is we'll develop cell cultures. I apologize if this slide is a little bit difficult to see. Sometimes the black and white is hard, but I wanted to share a picture of what these cancer cells actually look like. And so I think the best shot is on the upper left, which is an example of DIPG, diffuse intrinsic pontine glioma, which is a nearly uniformly deadly cancer that affects young children. And it's a good example of one of those cancers that just doesn't have a lot of good treatment options, lacks research. And so it's really exciting to be able to develop these cell cultures to study within our own studies and to share with other research groups. Down at the bottom, I just included a nice example of an osteosarcoma that we processed. and we're able to run a drug screen on drug screens. So this is what the readout of a drug screen looks like. I can talk through just a little bit. Across the top are the different samples, and then along the side here is 60 drugs. And this looks like a bit of an older drug screen. I think this was our version 3 drug screen. We're up to version 5 now, and we're thinking about building version 6. And so it's a smattering of drugs that affect all these different pathways on the far left. And the pathways are known to be oncogenic pathways. And then reading the heat map itself, the dark red would represent a lot of cell killing, and white represents no cell killing. So if you see the dark red, that's a drug that worked against that particular disease. And I included this one because it does have some osteosarcoma cells involved. And so there's examples that if you look deeply at this, you'll see that there's some good cell killing against the osteosarcoma samples. And then over here on the left are like normal cells, so osteoblasts. So in an ideal world, you'd want to see something that is really good at killing osteosarcoma cells, but is not good at killing a normal human osteoblast. That would indicate that it's probably the drug would be toxic to the tumor, but non-toxic to a normal tissue. I mentioned we also do a deep dive genetic analysis, and this is just a visual representation of a genetic analysis. We call this a circles plot. And around, just real briefly how to read these things, around the outside, the numbers represent the chromosomes. Of course, we have 23 chromosomes in a human. And then these little black lines that you can barely see, those are the genes that are located on the chromosome. So there's literally hundreds of these things when you zoom way in. So those are the genes. And then this outside ring represents expression levels. And so when something is very darkly red, that gene is severely underexpressed. And if it's very darkly green, then it's severely overexpressed. And then all kind of shades in between. Moving in towards the center of the circle, this ring here represents copy number gain or loss. And then the innermost ring is genetic mutations. And so every gene that is mutated is denoted here with a little red dot. And then on the very inside, these lines represent gene fusions. And so many sarcomas are gene fusion driven cancers. And so fusions become really interesting to us. And as an example where this one loops around, it would be like one gene fused up to this gene up here. So that's kind of, what we do with a lot of the samples. Where do the samples come from? All over North America. So we have relationships with institutions, hospitals, pathologists, all sorts of people involved in the treatment of children with cancer. And oftentimes they're aware of the work we do in the Cure Fast Tumor Banking program. And when they have a patient that's interested in making a donation, they'll reach out to us. There's also, just to point out, we also get introduced to patients directly by families. So I didn't include any of the families on here, but that's another source of these tissue donations. So now I'm going to pivot a little bit in the talk, and we're going to talk a little more in detail about autopsies and legacy gifts. Of all the logistical steps involved in a tissue donation, the autopsy is oftentimes the biggest obstacle, and it's for a variety of reasons, all of which can be addressed. Some of the barriers to autopsies include a perceived lack of research opportunities. So, if a doctor has a patient that speaks up and says, hey, I'd really like to donate my tumor, oftentimes the doctor doesn't know where to send it or doesn't know of groups that are researching the disease. So there's a perceived lack of research opportunities. we say go ahead and send it to us, because we study a lot of these tumors and we're very involved with the community. So we're aware of other groups who are studying all kinds of tumors. There's assumed objections, okay? These would be things, you know, that you just sort of make an assumption that the family is not interested, that they have too much going on, overwhelmed, and you know, sort of the last thing a doctor wants to do is have yet another difficult conversation. And so it's easy to assume that there'll be objections to it and not bring it up. Logistics. Like I mentioned in the beginning, the logistics of an autopsy and a tissue donation can be quite complicated, especially on the hospital end of things. We have sort of streamlined the process and think of it as a very simple, stepwise process. But oftentimes I'm speaking with doctors who have never done this before, and they ask me, you know, what do I actually need to do? And oftentimes my suggestion is, well, you need to take a walk down to pathology and have a chat with some pathologists and see if we can find one that's willing to do the autopsy. There's misconceptions about the autopsy. Namely, you know, the cause of death is known. We know this child died of cancer, so why would we do an autopsy? Grieving parents reluctant to consent. This sort of goes back with assumed objections. And disruption of funeral arrangements. You know, oftentimes children who are on hospice, they want to leave the hospital and they want to go home. And so there's a big logistical sort of juggling of what's going to happen when this kid does pass. You know, typically they would be moved to the funeral home, but now you want to do an autopsy to collect tumor tissue. How does that work? And the CureFast program is pretty unique, I think, in that we have this process figured out and we have funding to pay for body transport. And so it's a very, very simple process from the family's perspective. And so it involves the child, when the child passes, the family can spend as much time with the body as they need. We absolutely do not pressure them in any way to sort of speed the process. And so they can be with the body for as long as they want. And then when they are ready to let go, they call the funeral home. And the funeral home picks up the body and then drives it to the pathology lab. And we can pay for this body transport. So then once it's at the pathology lab, they do the autopsy, they send us the tissue, call the funeral home comes, picks up the body, and then drives it to the funeral home, and a normal funeral proceeds. So from the family's perspective, there's very little disruption of funeral arrangements. Autopsies in particular, you know, it goes back to the argument of why do an autopsy? We know the cause of death. Autopsies are big and long and complicated and expensive. So is there a justification for doing autopsies? We are big proponents of what I call rapid autopsies or limited autopsies. And these are autopsies set up only to collect tumor tissue for research. So it's not looking at the cause of death. It's a much simpler autopsy. You pretty much know where the tumors are, and they just go in and collect them. And oftentimes this can be done start to finish in less than an hour. Even faster than that is what we call a post-mortem biopsy. So in this case, they're not even, you know, opening the body up to get tissue. They're just collecting biopsies. So super fast and easy. Some benefits of an autopsy, multiple benefits. Just to medicine and science alone, autopsies should be performed because they're an excellent teaching tool. You know, as doctors and attendings and fellows and people who are on their way through medical school, an autopsy is a golden opportunity for them to practice on a real body, and that's very important. It also lets doctors and pathologists actually see visually the effectiveness and consequences of cancer treatment, which is important. And then, of course, the tissue is very useful for research and the development of study models. Benefits to the family. It's a positive outlet. It's a way to make something positive out of a tragic situation. It provides a sense of closure. It can honor the child. And it restores a sense of control. And anyone who's been through cancer treatment knows that that's one of the really horrible things about cancer treatment is any sort of sense of control of the situation is immediately stripped from the patient and family on so many levels. And so I always am giving advice of anything that you can control going through cancer treatment is a really positive thing. So being able to control what happens to your tumor tissue is great. The tumor is finally gone. I've had many conversations with patients and families that they just want that tumor out of there. And even in death, they would rather not have the tumor in the body. So this provides a great way to do that. And then I've also had many conversations with patients and families, and many of them are really interested in and happy about the opportunity to help other children with the same disease and maybe improve outcomes for future patients. Just a couple of slides on things to consider about discussing an autopsy. And I realize that many people watching this are not physicians and may not have to have, these conversations. But I also recognize that as a member of the childhood cancer community, you do meet other people and you may actually have a conversation about tissue donation or be asked about it. And so I thought these next couple slides would just be educational in case you do find yourself in that situation. So namely, the big thing to remember is this is a hard conversation that's difficult and should be considered delicately. So an important decision at a difficult time. One thing you need to do is consider your own feelings about the situation and recognize that the family or patient's feelings might be different, and you have to respect those opinions. The time might be right to have the conversation when a child is having a procedure done early on in the cancer treatment process. And we don't have to talk about autopsies in particular, but you can plant the seed early that, hey, research is really important, and this is a cancer type that does not have a lot of research, and that leads to not very good treatment options. And so just sort of planting that seed of an idea is an appropriate time to do it. Of course, when the family brings up tumor donations or asks if they can participate in research, that's a fantastic time to talk about tumor banking and tissue donation. Maybe when hospice is discussed, this is a little bit delicate and you just kind of have to feel out the family. But oftentimes when they're arranging for hospice, that's when they're thinking about what else can we do, and tissue donation might be appropriate. Always, advanced planning is key. This is not to say that we haven't pulled off tissue donations with just a few days' notice, but those are always very hectic, and I'm always holding my breath that things work smoothly. And so the earlier we can get into that discussion, the better. The time might be wrong to have these type of conversations when the child is initially diagnosed. So at that point in time, you want to have as much hope for the future as possible. And we probably don't want to discuss autopsies at that point in time. When the family is told all curative options have been exhausted, this is a very traumatic time for the family and very overwhelming. And so if it hasn't been discussed at this point, right when they find out that they're entering into hospice is probably not the most appropriate time, but there might be a window when sort of arrangements have been made to have the discussion. And finally, when the child dies, pretty much at this point, it's too late for an autopsy legacy gift, and we don't want to burden the family with these details. And rarely would it work at a donation at the last possible minute. The logistics are just too difficult. So with that, kind of brings me to the end of the presentation, and I'll just leave with this testimonial. This is Alexa again, and her mom wrote a very nice testimonial about her legacy gift. I know it's very wordy. I'll just leave it here in the slides to share, but I'd highlight some of the quotes. At the end of Alexa's time with us, I started thinking about her legacy and how I wanted to remember her when she was gone. It gives me great comfort to know part of Alexa is living on to help others. And finally, it makes me feel a little better to know that I'm part of the work to make childhood cancer universally survivable. So with that, I'll say thank you for attending the talk. And thank you to our funding partners, MIB Agents, for hosting this event. The Sam Day Foundation, which principally funds the Cure Fast program, Maddie's Promise, and of course the Childhood Cancer Project, which funds an aspect of Cure Fast that's related specifically to osteosarcoma. And then of course at the bottom here is our contact information. If you have any questions or you need to introduce a family to us, I'm available 24 hours a day, and you could feel free to share any of my contact information, and also my scientific director, Dr. Charles Keller. So with that, I will take any questions.
Thank you for that presentation, and thank you to all of those partners that make this possible. Because as an individual and as a family that has a child who's passed from cancer or is about to pass from cancer, this is a really expensive undertaking, really cost prohibitive, really, right?
Yes, absolutely. It's a big program, and there's lots of lots of moving pieces that are involved, and of course that translates into an expensive program, but absolutely necessary.
Yeah, I mean, really, really makes such a difference. It's when we have these programs, they're end of life missions when a child enters into hospice care, and 9 out of 10 times, I've shared this with Charles before, when we contact that child and say, what can we do for you? What would you like? What can we do? The number one thing they say 9 out of 10 times is find a cure. Nobody else should have to suffer like I have suffered. And then the second thing that comes up is, I want to do something for somebody else. I want to make it better for somebody else. And this is a way to do it. And in our experience with this program and with the kids who are contributing and wanting to contribute, it's driven by the child. It's been driven by the child or the young adult who said, I want my life to mean something. And right. Like Alexa, I mean, thank you, Alexa, for your gift, because that's helping future generations of osteosarcoma kids. And it's truly giving of yourself. And it's so profoundly meaningful to people who are in the fight now and who are yet to be diagnosed. It's so meaningful. So I'm so grateful to the families and the kids who make this gift. It's truly a gift. It's like that quote. It's something like, it's a Greek proverb. It says, thank God for the old men who plant trees and whose shade they will never get to enjoy. And that's what this is. It's these, not necessarily old men in this case, but young men who are planting trees, and they have no hope of enjoying the fruits of their gift. They're doing it for everybody else, really. So
yeah, I love that quote. That's wonderful, and it really nails it. That's exactly what it is. And I'm always amazed. I'm always speaking with the kids that are making the donations, and I'm always left with a sense of awe about how selfless they are. Here they are, sort of at the end of life, and their main thought is, how can I make it better for other children with my type of disease?
Right.
To hear that, coming out of a young teenager is really inspiring.
Really.
Right. I'll just add something that follows on this, the whole idea of the selflessness. I mean, it's so true. And the other thing, though, to consider is this is a really stressful time for the patient, for the family. There's lots of ways that these kinds of gifts could be done that would be stressful, but we've worked really closely with families to assess all the different experiences. And so we try to make it really simple. In fact, most hospitals do. You first get a single point of contact to talk to, like Andy, and then afterwards, you don't even have to worry about it. At some point down the road, all the doctors and Andy or the researchers are in coordination. You spend all the time with your loved one that you'd like. There's really no limit. There's zero rush. There's no reason not to sort of say the goodbyes in the exact way that you want to say goodbyes. And then at no cost and no stress to the family, the research procedures happen. Depending on how the preparations are for afterlife viewing, you won't even know it happened, right? Great, the child will have been liberated from the tumor, and yet you'll still be able to see your child, teenager, adolescent loved one. It's meant to be a seamless and stress-free procedure that is just an option to create a legacy.
All right, so the parents really have nothing to do until just signing the papers, and like once the child passes, they don't have to say, oh, I have to call you.
Yeah, that's absolutely true. We try to arrange all the logistics in advance. And like Dr. Keller said, we prefer to work sort of behind the scenes on purpose so that we don't cause any extra stress for the family. So it's done in a very smooth way.
All right, and then you listed several hospitals that are affiliated with you. How do we get other hospitals to get affiliated with you?
Well, so we work with pretty much all hospitals all over North America. And so when patients come to us, we just ask, which hospital are you being treated at? And then we'll directly reach out to that hospital. So the list, shared in the PowerPoint was a list of institutions that we have relationships with, but we certainly can work with institutions that we don't have relationships with as well. But to answer your question directly, if you're at a hospital and you would like them to be involved with tissue donations, it's as simple as a conversation with your doctor, and you as the patient or person there speaking with the doctor can mention that you know about the Cure Fast program and that you would like the institution to become involved with that to create a nice, smooth pathway for these donations to take place.
Have you ever met a hospital that is not willing to give information?
Not out of the goodness of their hearts. Many institutions, and we work with hundreds of institutions. Many of them, as you probably know, are bureaucratic machines. And so there are some cases where there's hurdles that need to be overcome, and sometimes it's really difficult to overcome those hurdles quickly. But I've never met a doctor who didn't want to see his patient cured. And they all, in my experience, they've all tried very hard.
That I agree with. Now I have a two-tier question. Will families and patients get any updates about what happens to their tissue? And if any genetic information is discovered that could be beneficial to a family, will they be told, even a year later? Hang on.
Well, Amanda, that's such a great question. Andy, I can take that one just because it involves sort of medicine. So we do this all the time. We communicate results back to the family, and we usually do it in concert with their clinician, because some of the findings that we have, some of them were just in the patient's tumor, some of them might have genetic implications, and you need the clinician there to help interpret what the next step is for the family who might also carry a predisposing mutation.
Yeah, okay, thank you. And can we donate tissue while the patient is still alive? Like when they're doing a limb salvage or an amputation, to give it a slice of what they take or something.
Yes, absolutely. You know, this was a presentation on legacy gifts and autopsies, but we certainly collect tissue from just normal tumor removals, you know, including all those things you mentioned.
Okay.
So there's another way that, you know, sometimes I have people at the last minute who want to make a tissue donation, but the child is like in the process of dying, and you know, like I mentioned in the talk, it's just a little too late. But even in those situations, a lot of times we can bring a lot of solace to the family by saying, look, the live tissue donation is probably not going to work, but what we can do is we can go back to the stored tissue. And so oftentimes these children have had multiple surgical procedures over the course of their therapy, and that tissue is typically always stored at the hospital. And so especially when the patient passes away, you know, that stored tissue is not going to do the hospital or the patient any good. And so we reach out and request that tissue, and we can take that stored tissue and do all kinds of things with it, including genetic sequencing.
Thank you. And one last question.
Okay.
I'm sorry.
No,
All right.
it's great.
Is there a benefit to getting the tissue after the trial dies? And are the cancer cells still alive when the person passes?
Yeah, so that's a great question. There is a limited time, you know, that cancer cells will stay alive, but it's longer than you think. It might be multiple days. So after, you know, the patient dies, as long as everything is set up, then for an example, you know, sometimes these kids pass away, you know, Saturday night, and there's nobody working in pathology. And so we say that's fine. Just store the body, keep it in a cooled room. And then the autopsy doesn't actually happen until Monday morning or Monday afternoon. And then it ships to us, and the tissue actually doesn't get here until maybe 4 days after the patient passed. And we still process that tissue, and sometimes those cells are still alive and they survive.
Wow. Thank you.
You're welcome.
If I can add something to that, you know, the remarkable thing is that the tumors of all things might be the most fragile in this process, but there are normal cells that we typically try to cultivate from the patient, and those are called fibroblasts. And those actually can be generated many, many days afterwards. And if you think about it, it's kind of neat, because not only may the tumor go on living and us learn from it, but the normal cells from that child will be also living in some way, also used in studies to understand the nature of the mutations that are responsible for the cancer.
Hi, I have two questions. The first one is, you mentioned that there becomes a point where it is too late to contact you guys for donations when in hospice. How much time is required to set up a legacy gift when someone transitions into hospice care?
It can vary, but you know, anywhere from like 2 weeks is plenty of time. I would say 3 or 4 days is maybe cutting it a little close to line up the logistics, but is possible. And I take these things very personally. So basically, anytime someone connects with me and is interested in making that donation, we scramble and we'll try to get it done in as quick as possible.
There have been many examples where or even though it was near the last minute, there was so much harmony between the family survivors and the physicians that it could be conducted. The situation that becomes harder is if it looks like the family or maybe the parents of the child aren't on the same page. We just say, you know, we'll learn something from another child. Please find a way to say goodbye that's comfortable for your family. Or if the hospital logistics start getting put on the family instead of all behind the scene, we say, hey, well, we could have learned something, but it's so much better for everyone involved if we just say, we won't do this one today.
Thank you. And the second question was, is extracted information also available on the CBio portal?
What a cool question. So sort of, we deposit our sequencing information in a number of public databases. So the tumor mutations can be found on dbGaP. We've done dozens, if not hundreds, of patient samples on dbGaP. GEO, which is another source, and EGA, which is kind of like dbGaP, but for Europe. I guess the long and the short is, every time that you have patient-specific information, you need to know that the person downloading that information is someone who's going to treat it as precious and private. And so while a researcher has to be registered essentially and agree to the ethical statements about using the information. So some things like CBio portal, which are fabulous in many contexts, are the right way to go, particularly because the people are registered, the researchers who log in to access the information.
But as a patient who maybe really wants their tumor data to go in a certain direction, say to cc-TDI, do you not have control over your own biodata, your own tumor samples to say, yes, I understand that the hospital needs a slice of this for their own record keeping or what have you, their own research, but do we not have an ability to say, and I also want it to go here?
Yeah, so most people honor the wishes about where your information goes and how it gets shared, and this happens all the time. You know, there was a New York Times article in 2011 for the It's My Data movement. So patients could create their own registries, like MIB Agents could create its own data registry, and the families would have direct, very specific control of where their information went, whether it was shared with other researchers, whether you want the information sold to a pharmaceutical company so they develop an osteosarcoma drug, right? That's actually a good thing. So what generally happens is the tissues are donated by the family, they're half the solution, and then an institution like ours or any university will sequence them, so there's a second investment there. And then the federal government that funds much of this research, or many aspects of research, will have the federal repository where information goes, and we tend to use federal repositories for the information. It's sort of an interesting question. Like, usually as a research subject, unless you control the entire process, you can't even know what your research data is. So tracking where it goes is a little harder. You know, people are adding Bitcoin to this so you can kind of follow that. No, it's true. There's actually endeavor out there to do that. So you can track where it went. But if someone's passed away and the data can be shared in a way that's respectful of the family and any inferences of inheritance, then you can be involved a little bit more specifically in your loved one's DNA sequencing sharing.
Right. I think people forget, including institutions, that HIPAA is not only a privacy document, but it's a portability document. That's the second P in there. And so you should have access to your records as a portable document that you can take and share with whomever you wish to share it with.
Right. You know, this is totally, you're totally correct. So there's two aspects of this. There are two levels of data: research lab and hospital grade. Hospital grade is called CLIA. Most of these studies that are being done are at the less expensive research grade. So it's not necessarily part of a medical record, and so wouldn't necessarily be something included in a medical record that would be transportable.
Mm-hmm.
But more importantly, there's even a few legal details. Like, so we're in Oregon. The state north of us is Washington State. In Oregon, a decedent is not considered a human subject. And so, you know, the consenting is a different process. In the state of Washington, the decedent is a human subject, and so that's a different research paperwork path. But in both states, the genetic information is considered covered by HIPAA because it has inferences to the family.
Mm-hmm. Right, I think as it should. Okay, last question for you. I think because we're about to be in overtime here. How do we make it better? How do we? I believe this is the way to make it better. It's my wishes when I pass that use my body. I'm done with it. Evidently, the driver has left the car. The car is available. What do you want? So I believe this is truly a way to make it better. To plant trees but how do we're so bad at these conversations about afterlife, about death.
Yeah, it's really a matter of communication and just, you know, the perception. So letting people know and also sort of conveying it in the right way. So there's lots of different things you can do after life. You can donate organs, you can donate corneas. That's not what we're talking about today. You can donate your body to science so that medical students can learn anatomy. That's not what we're talking about today. What we're talking about today is liberating one's body from the tumors, taking the tumors out very respectfully, and having all one's wishes and family's wishes honored so that the appropriate funeral services and send-offs can be done exactly the way you would want to do them without even knowing that the patient's been liberated, your loved one's been liberated, their tumors. And so it's really simple. We start out and we just say research is important when you're diagnosed, research is important when you're relapsed. And when you enter hospice, you know, research is still important. And one of the ways you can do that is the painless gift of tissue after life, which Andy had said. And if we say that, then it really demystifies and very accurately portrays this is the opportunity to liberate oneself from your cancers, to selflessly help others, and to have a respectful, peaceful passing for oneself and for one's family.
We need to know how to do this better. So thanks for sharing all of this important work that you're doing and giving us a way through to have this conversation. It's so hard. Okay, I'll let you know that next week, OsteoBites is taking a break, which is a perfect time to rewatch this episode and a perfect time to subscribe to our YouTube channel where you can view our library of 24 OsteoBites topics and OsteoBites rock stars. You can also listen to OsteoBites via podcast wherever you get your podcasts. So remember to visit mibagents.org/September to support our missions and programs for the osteosarcoma families that we serve. Please, it really truly makes an impact for the kids who are suffering greatly with osteosarcoma. It really makes a difference, I promise you. Thank you for joining us today. And of course, thanks to the Childhood Cancer Project, the Sam Day Foundation, yes, Maddie's Promise, to Alexa, and to the kids that we know, Patrick, Ian, Connor, the kids who have given this greatest gift to osteosarcoma kids still in treatment and yet to come. Their gift is so meaningful, and I'm so grateful to them. Thank you also to our guests, Andy Woods and Dr. Charles Keller of Children's Cancer Therapy Development Institute, our panelists and ambassador agent, Shannon McCormick, and OsteoWarrior and patient advocate Amanda Levine for using your powers for good Together, we make it better for osteosarcoma kids everywhere. Thank you for using your powers for good. Subscribe to our YouTube channel, and thanks for making it better for kids with osteosarcoma.